Welcome to the Functional Medicine Doc Talk podcast. I'm your host, Dr. Kurt Woeller, talking with experts in functional and integrative health and medicine, discussing critical information for improving your health and wellness to help you live a long, full life. Let's get to it. Hi, this is Dr. Kurt Woeller, and in this episode, I interview Patricia Kaufman. Dr. Kaufman is a PhD. She is the Dean of Integrative Health and Nutrition at the American College of Health Sciences. She's a certified nutrition specialist and licensed dietitian nutritionist with expertise in human health and performance. Dr. Kaufman authored a case report study of a woman with breast cancer who developed something called chemotherapy-induced diarrhea, which is a major problem for people undergoing chemotherapy for cancer, and how the use of dietary intervention and other natural remedies helped to improve the microbiome diversity that had been altered because of chemotherapy to greatly help this particular individual. And so this conversation is going to focus on the microbiome. We're going to look at chronic illness, particularly cancer, and how microbiome imbalances can be a contributing factor to underlying poor health, but also how microbiome diversity and the improvement of the microbiome can also have a profound effect on improving our health. We also get into important aspects of the microbiome, specifically looking at certain bacteria through bacterial classification, and get a deeper understanding of what are some of the compounds that these bacteria produce, like butyric acid, for example, which is a short-chain fatty acid, and the positive influences that have on overall biochemistry and human physiology. We'll discuss things with regards to probiotics, various lifestyle factors that are important for rebuilding the microbiome, and then, of course, just maintaining healthy microbiome diversity throughout our lifetime. So here is my interview with Dr. Patricia Kaufman. Patricia, welcome to the podcast.
Thank you. It's nice to be here.
So we have a lot to talk about. I want you to go just a brief overview of your education and your training, and then a little bit about how you got interested in the topic of the microbiome with regards specifically about chemotherapy and cancer. And we're going to have a conversation where we expand on that topic. But first off, go ahead and just explain a little bit about your education training and how you got interested in this topic.
Sure. I have a master's in nutrition, functional nutrition, and I also have a PhD in human health and performance. And the reason why I originally got into being more interested in the microbiome, it always starts personal usually, right? Why someone gets interested in it. And family members and friends that had, I have a number of family members that have Crohn's, number of family members that had cancer, colleagues that have cancer, and it started, and myself not having cancer, but always having an imbalance of my gut bacteria. And that could be because I was breastfed or I wasn't breastfed. I was given dairy as early as a child. I always had skin issues that was related to my gut. So that's kind of how I always, I got into being more interested. One is for myself and one is for others, to help others, because as they say, everything starts in the gut, right? All disease starts in the gut.
I think back on it, I was not breastfed. In fact, my mom told me that she was convinced by the doctors back then that formula was better. So I was formula fed. I never got breast milk. And I got introduced to dairy early on. I had terrible problems with chronic ear infections when I was a kid. And I'm sure I was on tons of antibiotics for that. So that was, still, a memory of mine too. So we're probably coming from the same,
Yes.
Same time frame. So you wrote a paper in 2024 in the journal Gut Microbes Reports. And this was a case study of a woman with breast cancer. And you specifically got into what you titled here, Preventing Chemotherapy-Induced Diarrhea and Microbiota Imbalances with Prebiotics and Probiotics. So let's first talk about that paper, the individual who was involved, because we know breast cancer is the second most common cancer in women in the United States. A large percentage of those women do chemotherapy. And this whole issue of chemotherapy-induced diarrhea is quite prevalent, and I know you're going to expand on that, and really getting into how the chemotherapy greatly disrupts the microbiome. And then, of course, in this conversation, we're going to expand on the microbiome because it's a problem that I think is really prevalent today in today's society and our world and the chemicals we're exposed to. And then maybe we can even expand this to perhaps other types of health conditions that people might consider even beyond cancer. But cancer obviously is a main thrust of this conversation. So tell us about that paper and let's start there.
So with those that have breast cancer, about 40 to 80%, it's a wide range, may have chemotherapy, will use chemotherapy as their first line of treatment. And with chemotherapy, the goal of chemotherapy, right, is to target fast-growing cells. And one of if you think of the gut lining, the epithelial lining, those cells are fast-growing cells. So when someone receives chemotherapy, and it goes with the esophagus too, right? Those are fast-growing cells. That's why those that get chemotherapy have a difficulty time eating because their esophagus is so raw, if you want to say. And so the same thing happens with the gut lining. There's damage to the gut lining of those cells. And so that starts leading to not only gut dysbiosis, but it also leads to the damage of those cells, which then causes lesions, basically, if you want to say, or interruption of those tight junctions in the epithelial cells that that we talk about leaky gut, right? And when leaky gut starts happening, that's when more inflammatory conditions start occurring as well. So with the chemotherapy, they're damaging those epithelial cells. They're also it also makes the chemo itself decreases really the contact time of what's going through the bowel, and that can lead to then diarrhea.
And go ahead.
And then I was going to say, and then because of chemotherapy, it can then target beneficial bacteria and increase the levels of pathogenic bacteria.
So are there specific cells within the microbiome, let's say the normal bacteria or the bacteria that typically live in our digestive tract, are there certain ones that are more vulnerable to the presence of chemotherapeutic agents in their depletion?
There are a lot of new studies coming out that are looking at, and I just peer reviewed a couple studies. One has just been published in the Frontier, looking at and it was on leukemia, can pathogenic bacteria predict that they might be at an increased risk of leukemia. And then another one was looking at what does the microbiome look like for those that have colorectal cancer. They usually have a decrease in butyrate producing bacteria. So they're bacteria that produce butyrate. So they ferment fiber, and then through the process, they they create short-chain fatty acids, and one of the short-chain fatty acids is butyrate. So butyrate is very protective to the epithelial cells, can prevent that leaky gut, that increased intestinal permeability, and that damage. So when the chemotherapy can decrease those bacteria, there is room for growth and a pathogenic bacteria. Like, we all have some pathogenic bacteria in us, right? It's really that overgrowth that starts causing the issue. And the proteobacteria are probably considered, one of the most damaging types of pathogenic bacteria.
So I want to get into the classification of bacteria. I want to do that in a second here, but I want to come back to something because I know this paper was on breast cancer. So this one individual had breast cancer, and you brought up the topic of colorectal cancer. Do you know if there is a pattern that exists within the microbiome that would occur linked to specific cancers, meaning that would in general, I realize we're talking about individuals, but maybe across the board, is there a different type of profile of the microbiome disruption that might occur in somebody with colorectal cancer versus breast cancer versus pancreatic cancer or some other type of cancer? Do you know if that exists?
It's all very similar. You have your decrease in your butyrate producers, you have your increase in your pathogenic bacteria. Like if you look at inflammatory bowel disease, they're more prone, their pathogenic bacteria are more E. coli, Klebsiella. You don't see that as much in those with breast cancer, but you could see some of the hydrogen sulfide producers. But you then see a decrease in the bifidobacteria and that decrease in the butyrate producers. And one of the things that is really key is to look at all of the bacteria. And maybe we'll discuss it later, but it's just not bacteria. You can have other, you can have fungi, and can have candida, you can have microtoxins, and even Lyme bacteria can cause an issue, right? But when it comes to breast cancer or colorectal cancer, it's very similar. There's one pathogenic bacteria called Volcatus, and that's a sign of like you have some inflammation going on in your colon. But you can't use that as a diagnosis tool, right? A diagnostic tool. But I do see that in those that I work with IBD, that they have that Volcatus is quite high. And that's a pathogenic bacteria as well. So and that's at the genius level. So I know we're going to discuss
we'll definitely get into that. And I wanted to just mention about butyrate, because you mentioned how critical it is as a short chain fatty acid. One of the things we know about butyrate is it also helps to support metabolism within the mitochondria. And that mitochondria can be within the digestive system, within the innate immune system, within the adaptive immune system, the brain, nervous system, all throughout the body. And so it's interesting that so much of the microbiome, at least maybe key players, have a fundamental role in butyric acid production. And we've often thought, at least in years past, when I thought about butyric acid, it was something that just acted locally. And I underappreciated its influence systemically, throughout the body. Do you know of having low butyrate, for example, over a long period of time? Is that a known risk factor for cancer development?
For any chronic disease development, really, is because one of the things that butyrate can do is it can promote anti-inflammatory cytokines. So it's helping reduce that systemic inflammation as well. And a number of people forget that 90% or 70%, it's like 70 to 80% of our immune system is sitting on the other side of that epithelial cell lining, the gut lining. And so when you have increased intestinal permeability, and then when you have low butyrate, it just opens the door up for pathogens to get through, and then the immune system to get upregulated. And you think about, that's the same with autoimmune conditions, right? How does, cancer doesn't start with one condition. Like there's just not one trigger, right? It's multiple things need to happen to increase the risk. And the same with IBD. IBD is, or any autoimmune condition, you're increasing the intestinal permeability. They have low butyrate producers. You then don't have your protection. You don't have your anti-inflammatory cytokine protection that the butyrate is actually helping. The butyrate producing bacteria are helping, because it helps promote immune tolerance.
So it's interesting. There was a recent paper that came out. I actually learned about it through Dr. McCullough in his newsletter. I can't remember the name of the article now, but it was talking about how complex 3 of the electron transport chain is often diminished in its function. And when it is diminished in its function, it actually has a relationship to macrophage activity, these big eater innate immune cells. And the deficiency in complex 3 activity appears to affect the ability of macrophages to transition from pro-inflammatory to anti-inflammatory cell states. So when we think of, and butyrate is one of those things that helps to rescue or improve the electron transport chain activity, including the relationship of complex 3 within that system. So the level of detail in molecular biology and cellular biology is amazing in starting to understand how these things work. But so much of it, and we were talking about this prior to the interview, when it comes to health maintenance, improvement, and overcoming illness, comes back to what's happening in the digestive system.
Right, and you had mentioned macrophages, and one of the role of lactobacillus. Lactobacillus is very small in our gut. It's about 0.01%. So it's a small player, but a mighty player. And one of the things it does is it helps reduce neutrophils and dendritic cells. And those neutrophils and dendritic cells are, basically other than macrophages, are our first responders, right, to start the immune process and the inflammatory process. So lactobacillus can actually, so if we have the right amount of lactobacillus, which is decreased as well in those that have some type of chronic disease.
Yeah, very interesting. So let's go through an overview of bacterial classification. So I even get confused with this because, and I've been looking at this material for a long time, but it gets confusing when you start talking about phylum and family and genus and species. And, I'm not trying to make this a biology class, for example, but, you do a lot of education and work with students. How do you describe and look at the classification of these bacteria and, from a general standpoint, and what's most important to look at when you're considering bifidobacterial lactobacillus and other types of things?
Yes, so the bacteria class or kingdom is pretty similar to the animal kingdom. You have different, I don't want to say classes because that's a word that's used in the kingdom classification, but it starts with phylum, and then it goes to class, order, family, genus, and species. When you're looking at we're just talking bacteria right now. When you're looking at bacteria, you really want to be at that species level. Why? Because the level above it is genus level. Well, in the genus level, you can, let's take, for example, Clostridia. You can have beneficial Clostridia and you can have pathogenic Clostridia. So if you only are measuring Clostridia and you're not looking at the species level, you're not going to pick up the pathogenic bacteria, or you're not going to know, do I have pathogenic bacteria or do I just have beneficial bacteria? And that's the same with the Bacteroides too. Like you can go down the class, the genuses, and there's some beneficial and there's some pathogenic. So unless you really can pick up that species level, you don't really know what you're working with.
One of the things that I've seen a lot on stool testing is they'll actually give Clostridia, as at that genus level. But again, they're not necessarily picking up on species. And I think it gets oversimplified. and people start to assume that Clostridium bacteria are all bad. And as you described, and I believe, Patricia, correct me if I'm wrong, about 40% of our microbiome is of this Clostridia bacterial genus. Is that correct?
It's a large number. And if we look at the butyrate producers, we were just talking about butyrate producers. Guess what? The Faecalibacterium perniciae, which is one of our main short chain butyrate producing bacteria, is a Clostridia.
Right. Interesting. So, that's the way I've always viewed it. It's like this is a very important I'll call it classification, but group of bacteria that play a fundamental role from a health standpoint within our digestive system, but there are some really bad actors.
Yes.
And those bad actors, if you look at just Clostridia difficile, can create a lot of havoc. And we know that it can create problems of inflammation and Clostridia difficile-induced diarrhea, and in severe cases, pseudomembranous colitis. And then there are other Clostridia that are bad actors, which we know can produce compounds that have an adverse effect in the brain and nervous system, which is where the organic acid test comes into play. What about Bifidobacterium? So you mentioned about lactobacillus, because when you go to a health food store, you pick up a bottle of probiotics, it's usually going to have some lactobacillus and Bifidobacterium. But there's been a lot of discussion lately about the depletion of Bifidobacterium in the digestive tract for various reasons. So where does Bifidobacterium fit into this discussion? And how concerning is it from your perspective of seeing such a depletion of Bifidobacterium in so many people?
So when we're born, so babies have about 60 to 70% Bifidobacteria when they're born. Those that have optimal levels, it's sitting between 2 to 5%, 4% plus or minus 4%. So I've never seen anyone with 8% Bifidobacteria all the time that I've looked at stool testing. Most people, and I'm not working with the healthy population because otherwise I'm not doing stool testing, right? So it's
right.
Usually, so it's the most of the people, clients that I'm working with, they're down at 1%, maybe 0.05%. They still have some, but they have very little. And so you really want that 2 to 5% is really the number you're shooting for. They're gram-positive bacteria. So if we think of pathogenic bacteria, they're what's called gram-negative. So that negative side of it means that they produce lipopolysaccharides, LPS, and those are toxic, right? So in the case of Bifidobacteria and Lactobacillus, they're both gram-positive. So you don't have that mix of, I have a genius level where I have bad and good. You pretty much have all beneficial bacteria in that for the Bifidobacterium. It improves eczema. So, of course, I do not have good Bifidobacteria. I've had a long life of eczema. It's much better now because I really work on my gut bacteria. It produces B vitamins, so that's another reason why. When you have low Bifidobacteria, it means you typically have more increase of toxins and more you have increased intestinal permeability, so leaky gut. So there's a negative association there. So
with a drop of Bifidobacterium, there's going to be likely an increase of gram-negative bacteria.
Correct.
Just maybe as I don't know if it's a counterbalance or sort of a compensatory mechanism or whatever.
Well, and if you think about it, we only have so much space we have a lot of bacteria, but we have only so much space. So when the beneficial commensal decrease, it leaves room for the pathogenic ones to grow.
That's a great way of putting it. There's only so much room. And then we get the expression of lipopolysaccharides, which is a triggering of mechanism or chemical for inflammation. not only locally, but systemically. So, and this is coming up a lot with just dysbiosis in general, with the overexpression of gram-negative bacteria. Is there something unique about lactobacillus, these gram-positive and bifidobacterium, of why they get depleted? I don't want to say easily, but it kind of appears that way, of why they get taken out.
Antibiotics.
The way they do.
Antibiotics. Stress. Because stress can cause the overgrowth of pathogenic bacteria. The toxins, chemicals. You name it. What just recently came out, we eat a credit card of plastics a week, which maybe is a little high, but still, right. Think of all the toxins that we have going through us. So you could say that they're a little bit sensitive, right, to toxins and to antibiotics. Even natural antimicrobials at really high dose for a very long time can target, because they're non-selective, and they can go after lactobacillus and bifidobacteria as well.
So are the gram-negative bacteria? I mean, there's
they're
normal
gram-positive. They're gram-positive.
Yeah, those are gram-positive, but let's say the gram-negative bacteria. I mean, there's some normal gram-negative bacteria in our digestive system that's
Correct.
part of a microbiome. Are they as sensitive to the environmental? Because you wonder why it's not going the other way. Right? Why aren't the gram-negative normal bacteria?
That's a good question. I don't know, but they're not as vulnerable.
I wonder if it has something to do with the cell membrane, for example.
Probably. Maybe that's why they produce LPS, to protect themselves. That's just a tip of mine. I would have to look into that, but that's interesting
thought though. So you mentioned about proteobacteria pathogens in the paper. So you talked on one thing I want to mention too. In the paper, you mentioned alpha diversity. You mentioned butyrate producing bacteria. So we've covered that. You briefly touched on proteobacteria pathogens, and maybe I want to come back to that again. And then just also the alpha diversity. What do you mean by alpha diversity?
It's a tool to measure the what's called the richness and the evenness. And so there's a number of different one. The Shannon index is a common one. And it's looking at how many players do you have and how many of those players do you have. So, and of course it's looking really at the beneficial, the commensals, than it's looking at the pathogenic, but it will still look at those. But what I see with a lot of chronic conditions is that alpha diversity score is very low. They don't have a lot of players, and the players they do have are more pathogenic, and there's a lot of them. So like you were just talking about, like, what does it look like when someone has a chronic disease? That's what it looks like. They have very few players, but those players they do have that are pathogenic, they have a lot of.
Now, is that the proteobacterial bacteria pathogen category?
In most cases. Like in some stomachs, it can be the proteobacteria, or in some individuals, it can be up to, I think I've seen it as high as 20% the proteobacteria.
And examples of proteobacteria are what?
E. coli, the hydrogen sulfide produces, so D-sulfuribyl, Bilophila wadsworthia, Klebsiella, even H. pylori fits in as a proteobacteria. So you can see those quite high.
Are all of those gram-negative?
Yes. Yeah.
They are.
Some are not as, less are known about all of them, but some are believed to not be as toxic. Their LPS that they produce is not as toxic.
So I think the key thing I want people to understand is it's the LPS component that there's likely other components, but it's that LPS component of a gram-negative bacteria that in too high amount can be very, very damaging.
Yeah.
Both within the gut and systemically in the body from an inflammatory standpoint.
Right. And we're seeing a neural inflammation being triggered by LPS as well.
Right. In fact, one of the things that LPS can trigger is microglia activation. Mm-hmm. So, and that's huge in obviously neurological conditions. We see it as a big problem in autism. So when you get an overexpression of gram-negative bacteria and you're getting a depletion of the butyrate-producing bacteria, so bifidobacterium, now all of a sudden you have a drop in this compound that helps with mitochondrial activity and also helps to prevent against maybe runaway inflammation. That would be the butyrate. And you're getting an overexpression of lipopolysaccharide, which is pro-inflammatory and immune triggering, particularly in the brain, those microglia cells. Now we have a real problem, and we can see where you can take really any chronic health condition, autism, chronic, neurodegenerative condition, cancer, autoimmune, whatever, and bring it back to the gut. So of why it's so important to focus and understand the relationship of the gut and the digestive system as a whole to our overall health maintenance. So with that, prebiotics and probiotics. So yes. There's a lot of people who run out, they'll just take boatloads of probiotics and think they're good to go. So first off, prebiotics. What is a prebiotic and why should people care about what that is?
So a prebiotic is basically the fuel for our bacteria. And so they're fermentable fibers. Not all fibers are fermentable. So the bacteria basically ferment the fibers, and those fibers are their fuel for their growth. So if you're going to pick over a prebiotic versus a probiotic, I would do a prebiotic because you want to grow what you have in you endogenously. For instance, if we wiped out our lactobacillus from too much antibiotics throughout our life, maybe we don't have any more lactobacillus left in our gut. And that can happen. Things can wipe out a whole genus and species. And so say we have no lactobacillus. We cannot take a lactobacillus probiotic to grow that lactobacillus that we lost, that we were basically born with. So probiotics do not seed in our gut. They're transient in nature. It doesn't mean that probiotics aren't useful, because when they are transient, they go through us, right? They do have signaling potential. So they signal to other bacteria. They signal to butyrate producing bacteria. So they're useful, but they're not going to grow. So if I'm trying to grow bacteria in a gut, I'm going to use prebiotics. And there are, my favorite prebiotic is sunfiber. Why? Because it doesn't cause digestive issues like a GOS or a FOS type of sugar, which is a prebiotic too. That can, if someone has a lot of bloating and gas, that can cause just more bloating and gas. So,
and FOS is the fructooligosaccharides.
Yeah.
When we're born, right, we have implantation of bacteria. We should coming through mom's birth canal, and then breastfeeding on top of that. What is it about the newborn digestive system that allows for seeding to take place? I guess the way I look at it is like you seed and then you grow roots that a probiotic wouldn't be able to do over time.
Well, no. So we really are born with our bacteria. Because I said 60 to 70% bifidal, right?
Yeah.
Well, there's that 30 to 40% that's something else. So, and remember, right, lactobacillus is so very small, 0.01%. So we pretty much have the bacteria. And so when we're not breastfed, like you and I, we can have difficulties we might not have some bacteria that we should have.
Wow, it's almost like we're doomed from the get-go from back
Yes.
at that time, Whereas I know, kids today, I mean, our kids got, breastfed for a couple of years. So, it wasn't by me, but obviously it was, my wife did and was really dedicated to it, so some people might argue that was too long, but it was clearly a concerted effort, to make that happen. Whereas opposed to our generation, it sounds like at least wasn't, there was clearly a propaganda campaign for formula over breast milk. So you like sun fiber as a prebiotic. I often think of prebiotics as fertilizers for the good bacteria. That's the way I've sort of thought of it for myself. Are there how else would somebody work at building their microbiome naturally through diet? What are some other things that you like from a dietary standpoint?
Polyphenols. The gut bacteria will use polyphenols because we don't really, think of foods that aren't absorbed by our body is what the gut bacteria like to use. So fiber that's not absorbed, right, that they can ferment and use for their growth. The same with polyphenols. Polyphenols have a very poor absorption rate, right? And we do a lot of things to try to increase that absorption rate, but the gut bacteria use polyphenols for their growth. So a plant-based diet is very beneficial.
Right.
Blueberries. I was just doing a Meet the Deans, a dean at deans at our at American College of Healthcare Sciences, and one of the students asked, what holistic nutrition practice do you do every single day? And I said, eat blueberries. Because, there's a great PDF, and I can share it with you, from the University of California, San Francisco, so UCSF. And it's on breast cancer, and it's what foods to eat to prevent and to help while someone is going through breast chemo treatment. And one of the things that they recommend are blueberries. And they've shown that blueberries can actually decrease the risk of breast cancer. This particular one was showing breast cancer. So blueberries, the bifidobacteria, all the short-chain fatty acids, the butyrate producers, they like blueberries. Arona berries are very good for acromansia. We didn't really talk about acromansia, but they, at adequate levels, acromansia are very beneficial for us.
So years ago, I was at a medical conference in my home state, and it's a pretty traditional medical conference, but they had pretty much a half a day on the microbiome. So somebody was talking about microbiome of the gut, microbiome of the skin, microbiome of the mouth. And one of the biggest take-homes I found from this one nutritionist that gave a great talk was they looked at a study of all of the different variables that had the greatest impact on the microbiome, from exercise and sleep, et cetera. And it was eating a plant-based diet. The average of trying to consume between 12 to 15 plant-based foods per day. And that really struck me. I actually took a screen capture of that slide, and I use it in some of my lectures. And right after that lecture was lunch, and we walk out and there's this beautiful buffet of just, plant-based foods. There was some meat there too, but that really stuck with me. And so that, I think, is a major mechanism. So I'm going to ask you a question. This wasn't on my list of questions to ask you, but I'm going to ask you anyway. And what is your take on something like a carnivore diet? From the standpoint of the microbiome, because you're hearing a lot about it now. And I've been around a long time, Patricia, as far as the different diets and diet fads and whatnot are out there. And I'm not claiming one thing one way or another, but my personal, experience from a general health standpoint long term is to sort of think about traditional way of eating that would incorporate a wide variety of food, and certainly with a lot of plant-based foods. I mean, I'm a firm believer, from not only for myself and what I've seen, something more like the Mediterranean, their longevity diet, et cetera. What do you make of the carnivore diet from a standpoint long-term with regards to the microbiome? And maybe you have some experience with this.
So one of my roles is I'm a CNS supervisor, right? So I review basically SOAP notes from nutritionists that are working towards their CNS certification. And one of the candidates that I work with, she works with a number of clients that are on the carnivore diet. And every time I review it and I give her comments, I'm like, gut dysbiosis, because they're not, their body, they have no, and even their symptoms are showing gut dysbiosis, right? And their food intake is void of plants. And those plants, think of legumes as plants too, and whole grains, that all of that helps really feed our good bacteria, our beneficial bacteria. So when they're not consuming any of that, you have to think they have no butyrate producers, they're probably really low in their bifidobacteria, and they probably have an overgrowth of pathogenic bacteria. And then
right.
They're on the carnivore diet. Why? Because they want to lose weight. But if you don't have beneficial bacteria, that you're not going to be losing any weight. So,
well, I think a lot of people go on it too because they have a lot of food sensitivities, which could be driven by other things. They can be driven by other dysbiotic factors than things that are leading to mast cell activation. So you have to start digging deeper as to why somebody would get benefit from a diet like that.
Right. And, and a number of people, we can shift our bacteria pretty quickly. So if you eat a carnivore diet for 1 week and then you switch to a plant-based diet on next week, you could probably see a good growth in your beneficial bacteria and a decrease in your pathogenic bacteria.
Yeah, that was one of the things that always intrigued me about the digestive tract and the microbiome is how quickly things can shift, which really shows you from a chronic health standpoint, if people just start improving diet and just improving the quality of food, they can see positive changes pretty quickly. Part of this too, when you mentioned about the phytochemicals, I recently read a book called What Your Food Ate, and it was all about soil and the health of the soil and getting to aspects about regenerative farming and looking at soil health and how that translates into the abundance of nutrients in foods beyond just the macronutrients, and really starting to look more at those phytochemicals and many compounds that aren't even really truly classified into, is this a vitamin or what is this substance? That there's so many of these different types of phytochemicals, I think, that are still to be discovered. So, I've often looked at the microbiome of our body. Is equivalent, maybe in the other way, is the soil of our planet and the health of our soil. The soil is the microbiome of the earth. And our microbiome is obviously, the beginning stages of the soil of our body.
Well, you had asked previously, what is one reasons why someone has gut dysbiosis or an imbalance of gut bacteria? And the other is the hygiene theory, right? Is we're too clean. And we used to play in the dirt. I'm sure you did, as I did. I'm from a big family, so we played a lot of times outside because get out of the house, get out of my hair. So, but you played in the dirt. People don't play in the dirt. They wash their hands. They're not exposed to, their body doesn't know how to deal with any type of pathogenic bacteria or infection because it's never been exposed to it. So there is that whole, germ-free theory or, that you're the clean theory. So like you said, as we deplete the soil of healthy bacteria as well, it does impact us as consumers of products that grow in that soil.
Yeah, the reality is we need our immune system to be trained.
Yeah.
And so some people think that the innate immune system has no training, but actually it does get trained. So there's not necessarily memory per se, but there's a training aspect to certain cells within the immune system. So natural killer cells, for example. What about the microbiome? So we're hearing about the microbiome. So the microbiome, at least from how it's described, most people think of it as a bacteria. And then there's the microbiome, so the fungus. Where does that come into the discussion? And clearly we can have fungal infections, but are these fungus even at a low level, perhaps as the backdrop of the greater microbiome discussion, actually having some beneficial influence?
Can the fungi have beneficial influence? Is that your question, or negative influence?
Yeah, I'm just because we know that Candida is a normal, organism within our digestive system. So it's a normal part of the ecology of our planet, of our body. And I'm wondering if there's any type of beneficial influence that the microbiome has. For example, it's not at a great percentage. We always discuss it from a problematic pathogenic standpoint. But I've often wondered, well, why is it there? And is it just solely being controlled? Or is it perhaps having some beneficial influence? I'm wondering if you know about that.
That's a good question. You're right. We all have Candida in us. And when you tell people that, they're a little bit surprised. They're like, no, I don't have any Candida in me. I'm like, But I look at more of the negative side of Candida. So when you have gut dysbiosis, even you give the chance of Candida to overgrow as well. So, and it's the same with mycotoxins, right? We could have some mycotoxin in us. It's the imbalance of our gut bacteria is set in the stage, right? It's just that overgrowth of the Candida is that one more needle on the haystack, or the hay that broke the camel's back. So it's just a compounding factor. Right. So we really need to look at our gut as a whole ecosystem. It's just not bacteria. It's just not mycotoxins. It's just not fungi. It's all of them. They all have to play together.
I like that, looking at it as an ecosystem for sure. What, in your opinion, in your experience, are some of the biggest environmental factors do you think is actually having a negative impact on the microbiome for people?
A number of things. Stress, right? Stress increases LPS. There is the plastics, like I said, the phytochemicals. Those can definitely influence it. A diet, right? Look at the environment of that, low fiber, high saturated fats. Nutrient deficiencies, right? It's a stress on the body. So any stress on the body can be an environmental factor. Over exercising, that's a stress on the body. Exercising can help the gut bacteria, but it can also play a detrimental role if there's too much exercise. So
do you know how that plays in? So is that a adrenaline type of effect? Is it a cortisol influence that's happening from stress or too much exercise? Do you know?
Inflammatory process, right? So It can then target beneficial bacteria and potentially let the overgrowth of pathogenic bacteria. So
years ago when I was in San Diego, I did a lot of work with a lab called BioHealth Laboratory. And their founder, Dr. Bill Timmons, was a real genius guy. He was a naturopathic doctor. And they had done a lot of studies on excess cortisol production just through hypothalamus pituitary adrenal axis dysfunction and linking it to immune suppression. And what they would often see is when cortisol levels would rise, you'd get a suppression of secretory IgA production. And so, lower secretory IgA, you're getting lower mucosal immune, activity. Obviously, you're going to get an increase, potentially anyway, in pathogenic organisms. But I have often wondered if the lowering of the secretory IgA had some direct effect on the positive bacteria, or maybe it was the cortisol that was having a suppressive effect. I'm not sure.
I would suspect it's the secretory IgA, because that's our first line on the mucosal surface, right, is our secretory IgA. So when you have a pathogen that's coming through food, like just say foodborne infection, right, a bacteria coming in from orally, something that you ingested, if you don't have the right amount of secretory IgA, which you're right, can be reduced from cortisol, you don't have that first line of protection. Now that pathogen could potentially get through and cause systemic inflammation. And then remember, your immune system is sitting right outside the other lining.
And I believe many of the bacteria that produce butyric acid would also stimulate or can stimulate secretory IgA production.
Correct. And as we spoke before, so can lactobacillus be beneficial?
Right. So it all cycles back. It's very, very interesting when you start thinking of the mechanisms of this, as so much comes back to that healthy diversity within the microbiome. So bringing it back to the core aspect of this discussion and the individual with cancer. So we've got the patient who had breast cancer and the diarrhea induced from chemotherapy. From maybe it's breast cancer, maybe it's just cancer in general, Patricia, are there certain things that these people should do or people in general should consider overall from a lifestyle standpoint? We've talked on diet. I know what you're going to say, plant-based diet, but overall, let's just kind of wrap this up of general recommendations from a general health standpoint, things that people can pursue if they have cancer, if they don't, they want to prevent, all the things that we can do to try to improve our health. What are some recommendations you'd make?
So in addition to a plant-based diet, which would be like a Mediterranean-like diet, right? Because it's high in polyphenols and fermentable fibers and antioxidants. So definitely looking at diet and also looking at their gut microbiota, their microbiome, looking at that and seeing how they, especially before chemo, they get diagnosed before chemo, checking out their gut bacteria and even everything, the candida, the mycotoxins, and addressing those, with prebiotic type of fibers. The other thing is with someone it's very important that someone that might consider some of the probiotics that we used in the case report is to always work with their oncologist team before they take any type of supplement, even prebiotic fibers. They should always work with their team first before taking any type of supplements. But looking at, if they are not going to take supplements, looking at food. But there are a number of probiotics that could help prevent the chemo-induced diarrhea. One of the individuals that I worked with colorectal cancer, she was doing long-term treatment of chemotherapy and before she came to me, and doing some of the probiotics, the different strains of probiotics really helped cut down diarrhea as an adverse event or effect. Is stage. So stage 1 means that they have 4 to 7 bowel movements a day. Stage 2 is greater than 7, stage 1 is less than 4. Stage 2 is greater than 7. I think that's what it is. But she had greater than 7. So she was at a high stage of having stage 3 and 4. They usually have to stop chemotherapy because they have such severe diarrhea. And so she was at stage 2, sorry. She had more than 7 bowel movements. And we were doing some prebiotics. So we were doing the sunfiber. We were also doing some specific strains that Saccharomyces boulardii, especially Florastor. And the reason why I mentioned Florastor, I don't like to always mention brands, but they use a specific strain, which is CM1-745, such exciting letters for strains or names for strains. That is Saccharomyces boulardii is actually a yeast, but a beneficial yeast. So you were talking about beneficial yeast before. So Saccharomyces boulardii can really help with that whole preserving damage to the gut lining, and it can really prevent pathogenic bacteria from adhering to that gut lining to then cause the damage. So Saccharomyces boulardii can be very beneficial. And then there's a number of other Rhamnosus GG can be beneficial for that gut lining, and there's a couple of bifidobacteria and lactobacillus that specific strains that can be beneficial. So taking those pre-chemotherapy could cut down on the risk of having that chemo-induced diarrhea.
Saccharomyces boulardii also stimulates secretory IgA production, and it has an inhibitory influence, at least with some of the pathogenic Clostridia, like Clostridia difficile. So clearly the chemotherapy-induced diarrhea is a big problem. I'm wondering, in your experience, it's recognized, clearly it has to be in the world of oncology in chemotherapy, but is what you're talking about as far as prebiotics and probiotics and eating a plant-based diet and trying to work on these things, is that appreciated, from a broader perspective?
Not with every, no, I would say. Well, and the reason why, it depends on your team.
Right.
My uncle had cancer of his uvula, right, up by the esophagus, back in the throat. And he was on radiation. And I gave him just a smoothie recipe, right, because everything goes down much rougher where you're having that radiation. And we did not do any pre or probiotics, but we did a lot of plant-based foods. And his oncologist loved it because he kept his weight. That was the one thing. They wanted to make sure he maintained his weight. And the radiation treatments were, he wasn't in as much pain from the radiation treatment, from just this plant-based, protein-rich smoothie. And so his oncologist loved it. Well, one individual that I worked with the colorectal cancer, her oncologist was like, well, you can take those, but I don't think they're going to work. So it really depends on the team and what they know about the gut microbiome.
So really it comes down to, like so many things, the patient advocating for themselves and people doing research and understanding, like podcasts like this and others, and information that you have to try to learn more. Because nobody in medicine knows everything. It's always a constant, process of learning and understanding. So what are some favorite lab tests that you like for assessing gut health and the microbiome?
I wouldn't say there is one favorite test that I like. I mean, the test that is really the gold standard right now is a metagenomics type of test. So it's testing the DNA instead of the RNA, because we really didn't discuss the different types of testing, but it's looking at fungi, it's looking at yeast, and it's looking at bacteria, where 16R RNA, which was used in that case report, it's more specific to the genus level, and less specific to the species level, where metagenomics is looking at the DNA. So it's looking at all the players. So is
that 16S ribosomal RNA technology. Is that what you're talking about?
So metagenomics is beyond 16S RNA,
Okay.
but the 16R RNA, when I do use that, I use BiomeSite. But it's only looking at 16S RNA. The metagenomics, it's come a long way in meaning that it's not as expensive, but it's still expensive. It's an expensive test, and it can take a while to get results. And like we said, your gut microbiota can switch so rapidly. So it's like, if I'm trying to figure out what's going on, in a short term, and I don't get the test back for 2 to 3 weeks, it's like, well, I don't even know if that's still the case anymore of what I'm seeing.
Right.
So that's the downside of the metagenomics, but they are decreasing that time that the test results are available. So,
are there specific tests that you do to look for pathogens, or is that metabolomics or metagenomics test assessing that too?
If it depends on the type of pathogen, right? If I'm looking at specific Clostridia, I really like the organic acid test. So I do use a lot of the mosaic test as well. My ideal test would be looking at the gut bacteria test, or even the metagenomics test and the organic acid test, because that could tell you a lot. And recently, I just worked with someone that they had both organic acid test from mosaic, and then they had the BiomeSite, so the 16 RNA. And, she's had long-term gut issues. And it's like, oh, well, she didn't realize she had so much Clostridia in her, the not so friendly Clostridia. So we could see that she had a lot of the proteobacteria increase, but we couldn't really gauge that Clostridia. So using the organic acid test really helped to shed light. And then she had candida. She had an overgrowth candida. So in my ideal world, there would be a combined test. So.
Oftentimes we need multiple tests to just be able to increase the view, the image, the three-dimensional image we're trying to look at, so to speak. Multiple tests give us different camera angles. Where do you think we are in this space of microbiome assessment, understanding the diversity of the microbiome and the different bacteria, even throwing in the microbiome, as far as the fungus? Are we still in an infancy stage? I mean, have we come a long way, or is there just miles more to go?
I would say we're a toddler.
Okay,
so
I like that.
We're not just at the end, there's some known, but there's still a lot unknown.
Right.
So they say we have maybe 4,000 species. We might know about 1,000, but out of those 1,000, we don't even know how to test those 1,000. And we don't even know what all the 1,000 are doing. So, we only just talked about a handful.
Yeah.
And now we're talking that we have maybe 4,000 species in us.
Right.
So,
I know it's crazy. Well, it's what's to come, right? And it's exciting. It's an exciting time, in medicine and healthcare, to be able to have these tools and to understand, but understanding there's still more work to do.
Right.
And it's so great to have you out there being part of the team doing this kind of important work. So some additional resources that you'd like to provide for people out there who want to learn more about what you're doing, this kind of, discussion and what we've been talking about for the past hour.
Resource. I mean, I have a website, which is purplegreens.life. I'm also the dean of ACHS. So if anyone wants to get further educated, they can always look at either the Masters in Holistic Nutrition or the doctoral program, which is in Integrative Health. And I'm more than happy to share with you that breast cancer nutrition guideline from UCSF. So
yeah, we can, and all of that too with your bio goes on your webpage. So do people contact you or learn more about you directly through, what was that website again?
They can either learn more about me through purplegreens.life or achs.edu.
Okay, so that'll all be in your bio. We'll make sure that's on the webpage. So, Patricia, it's been a pleasure to have this conversation. I certainly learned a lot. There's so much, so much to this particular discussion. I think too, there's a lot of confusion, and I think it's really important to understand in what you described, in that you're not going to replenish per se a faulty microbiome just through probiotics. It's got to, it really comes down to diet and lifestyle. And that's a huge component. I'm a huge believer in that. And it's something that I, try to get out there as much as I can through my own consultations that, again, it's not going to come down to just a simple probiotic. It's not that probiotics aren't important. They obviously are, but there's more work that needs to be done. So, well, thank you.
Thank you. Thank you for having me.
You've been listening to the Functional Medicine Doc Talk podcast with Dr. Kurt Woeller. For more information about this podcast, go to functionalmedicinedoctalk.com.